Hey, Kiara. Can you guys hear me?
Yeah.
Anybody? Oh, okay, good. Thanks, Elizabeth. I hate it when I'm in a meeting and it's like, I think I'm communicating and then nobody's hearing me. And so I'm just like shouting in the wilderness. All right. We're missing, oh, there's Kelsey. All right, everybody, unmute yourselves if you wouldn't mind. Like permanent unmute. Thank you. Okay, I want to spend a couple minutes here at the beginning talking about your time with Dr. Dilts. So this is something I've been doing for the last couple of years. It's a method that was developed at James Madison University, where Dr. Dilts used to teach before she retired. Dr. Dilts was the department chair when I arrived here back in 2000. And then she took a position at James Madison, and now she's moved back to the Liberty area because it's kind of where she feels like home is. And it's designed to give me feedback about kind of how the course is going mid-semester so that I can make course corrections, so to speak, as we go through the rest of the semester. And a couple of things jumped out at me from the comments that she communicated with me. One of the things is the whole thing about lecturing and the fact that lecturing sucks on Zoom. Lecturing sucks anywhere, but it sucks especially badly on Zoom. I'm working on some ways of being less lecturing. One of the ways that I did it was by asking you guys to do the family summaries instead of me just being responsible for all of those. Another one of the things that I did was when I asked you guys to think about what are the characteristics that would make good transoceanic dispersers, transcontinental dispersers, and human facilitated dispersers. And I don't remember what you guys remember of that process, but what were your thoughts as you were going through that?
I think what we talked about with the families was like we all thought it was beneficial.
Kelsey, are you talking? Because I can't hear you if you are.
Well, that's unfortunate.
I can hear Kelsey fine, so I don't know.
You. Wait, you guys can hear me?
Maybe there's something wrong with my audio. Can you guys hear her?
Yeah.
Ah, there must be something wrong with my video. Let me see. Kelsey, speak.
Hello?
No, I can't hear a thing. All right, hold on.
That's funny.
Try to participate, man.
Kelsey, speak to me now.
Hello?
Oh, yeah. Woo, yay. Yeah, technical difficulties on my side. Yeah. Oh, so what were your thoughts as you were going through that whole thing with biogeography?
Yeah, so I think what we talked about with like the biogeography in the families was we thought like we liked the contribution, like being involved in like that kind of thing, but we thought it made it a little more difficult to be sure of like what we needed to know, especially with the families, because one thing we talked about was just that it was like hard to write things down that quickly without having it on the slides. And so just kind of like not being sure what exactly we needed to know going forward, especially if like the test that had the family sections, that made it a little more difficult.
Yeah, so I mean, I tried to provide summaries on those family slides of the things that I thought were important. Were they incomplete in some way?
Well, we just weren't sure if we needed to know like things people said, or because like a lot of times the slides only had like 2 or 3 things, and then people would list off like a lot of things.
Oh, gotcha. Yeah. So, yeah,
That makes
certainly.
sense.
So basically, if I've taken the time to put it on a PowerPoint slide, chances are you should know it. I mean, because
Yeah.
I've taken the time to construct that thing. It's a little difficult when we do things like the family thing to know what's going to be said by the student. So, for example, presence of teeth on the maxillary was one of the ones that came across on the exam. I'm like, wow, that was not the character that I would have gone for that particular family. Now it happens to be true for that particular family where that was one of the answers, and I gave people credit for it. That wasn't one that kind of made my list of top things, because I tend to think about things that I would see on the creature if I was holding the creature in my hand. So, but 2 of you actually had that, and I forget actually which family it was. That's how kind of unimportant I think of that particular characteristic. But because you guys had it down there, and I actually had to go and look it up and see, is that really a characteristic of the family? Then I graded it as correct because that was the correct answer, even though it wouldn't have been my first or second or third or even fourth answer as to what distinguishes that family from other families. So what I've put on the slides is what I think are kind of the most obvious, most important things. And there were a number of things that I knew about some of the families that you guys covered, but it wasn't the thing that bubbles up in my mind as being the first thing that I think of when I think of that particular family, which is why I didn't have those things on the slide, is because they're substantially down in my list of things. They're interesting things about the family, but there comes a limit as to how much you can pack into your brain. So I've tried to use the slides to kind of distill what I think is most important. And so it's not to say that things that you add to those slides are unimportant. It just means that those aren't the things that I thought were most important. Does that help at all on that thing? So this is another thing that Judy mentioned to me, this idea that it's hard to know what you need to know. So I wanted to do a little exercise with you based on our conversation on Monday, which was only 30 minutes. We spent 30 minutes talking about water relations. What do you think I want you to know about water relations? There's also a good test review.
Like the 3 types of water, like the preformed water, metabolic water.
Liquid water, preformed water, and metabolic water. Yep. So I definitely want you to know about those 3 things, and I want you to know the details behind those 3 things. What other things might I want you to know about water relations in amphibians and reptiles, based on our conversation on Monday?
The water postures and how that like helps animals to conserve or get water.
Okay, sure. Water postures. And we talked about water postures in snakes. We talked about water postures in lizards. We talked about water postures in turtles, tortoises. Today we'll talk about water postures in frogs, which we didn't get to on Monday. What other things do you think I might want you to know about water relations based on Monday's conversation? This has to do with kind of the last topic that we talked about right before we broke.
Skin resistance?
Yeah, skin resistance and modifications to the skin that result in different organisms having different levels of skin resistance. And in particular, we talked about how squamates differ from, or how reptiles differ from amphibians, but then we talked about some interesting adaptations within amphibians that lead to quite a wide range of variation in skin resistance among amphibians. I think that's actually a pretty good summary of what I want you to take out of those 30 minutes. And here's the thing, I think it's pretty clear from the PowerPoints that those are the things that I want you to take out of it. Am I mistaken in that, I guess, is my question.
I think our thing was kind of like when you go off on tangents sometimes.
Yes, I am going to get to tangents. Yeah, go ahead.
That just makes it like we know the things that are in the slides are listed, but the tests don't just ask straight from the slides. I mean, we've all learned that since E&E. So I guess that's kind of the thing of like differing between when it's like you're just adding details that we do need to know, or when you're telling us a story to make it interesting. I guess we kind of like
Yeah. So
that.
generally speaking, if I'm adding detail that's not on the slide and I don't expect you to know it, I will generally say either right before or right after it, I'm not going to expect you to know this. And I do that periodically. Like, here's a little bit of extra information. You don't really need to know this. Stories that I tell, I realize I do tell stories a lot. The main reason that I tell a particular story is because it has relevance to illustrating something in the real world about the thing that we just talked about. I try to not go off on random tangents that don't have anything to do with the topic at hand. You could help me if after I tell you a particular story, if you just said, any of you could say this, in reality, you have total permission to say this. What exactly did that have to do with what we were just talking about? Because in my head, it's clear what the story has to do with and why it ties to the thing that we talked about. But I also recognize that in the process of telling it, that might get lost in the details. And if I have that reminder, like what exactly did that have to do with water relations, it might help. So for example, last time I pulled information that was not in your textbook from the paper by a guy named Brian Jennings on tortoise foraging. And I went into that in some level of detail. And what you might have asked yourself at the end of that is like, why the hell did he bring that up at this time? And spend four slides on it, actually. And does anybody know why I talked about the tortoise example, the tortoise foraging example?
You're basically talking about how tortoises will preferentially eat certain like plants that have a lot of water in them.
Yeah, so that came out, and I could have told that story without the benefit of added graphics, but the graphics were there in the paper and they were easy to understand, and I was able to find photos of all of the species that they seem to be selectively foraging for. And I wanted to get across to you this idea that the tortoises are really selective, and the things that they're selecting are things that either give them protein, which is important for the life of a tortoise, or give them water. And since we were talking about water relations at the time, and not only that, we were talking about preformed water, that was the purpose of that diversion into something that wasn't explicitly in your textbook, because quite frankly, that paper's been published since your textbook was published, and textbooks at their best are always 4 or 5 years out of date. So know that when I take those asides and I tell those stories that I, at least in my mind, think that they have to do with the topic at hand. If at the end of that it's unclear to you what the connection was, by all means ask me to clarify, like, what was the point of that? Because it might be clear in my mind, but it is not necessarily clear in your mind. But usually I tell those stories not because they're going to appear on the test. I tell those stories because they illustrate the idea that we're talking about in a real world kind of way. And if you would rather just not hear those stories, I can also just cut those things out. But I hope that they add to the topic rather than distract from it. But I think in most cases, I wouldn't even test you about those things. They're just illustrative, intended to kind of cement that idea in your head. What was the other thing that I wanted to talk about? Let me go check my notes. That's the wrong document. Oh yeah. So, well, I asked you about the biogeography task. So I put you guys all on this biogeography task. What are the characteristics of transoceanic, transcontinental, and human facilitated dispersers, and what kinds of organisms within herpetology, within the herps, would you expect to do this? And then I sat back and you guys worked for a while. What did that feel like to you?
I think it was okay for like independent learning, like for myself, but it wasn't really like a discussion in any way, or like working with other people.
Yeah, so it wasn't a discussion. So when I do this in class, and this is something that I've done in class before, that was not a new slide that I did specifically for you guys. It's something that I've done in the past. The idea is generally that you think on your own, and then you turn to one of your neighbors in the classroom and you talk about it. And then we discuss as a class, and we skip the talk about it in pairs. So at the title of it was Think, Pair, Share. It's a common pedagogical technique. A little more difficult to do online because you're not sitting next to one another in a room. So having that discussion amongst yourselves would be nice. It's a little harder to do on Zoom. Other thoughts about that exercise? Do you guys remember how long you went through that? Have you looked at that video at all? It was 15 minutes. I watched you on video and I could tell when you guys were kind of finishing up because your postures changed. You know what it's like on this side of the screen to just sit for 15 minutes watching you guys?
Boring?
What's that?
I said boring.
Oh my God, I'm crawling out of my skin. What would I normally be doing if we were in a classroom during those times? Because you've experienced these kind of protracted long periods of you guys working and the professors not doing anything in a sense. But when you've seen me do that in, for example, in evolutionary ecology, what am I typically doing? Am I just sitting up in front of the room, twiddling my thumbs, checking email? What's that?
Walking around.
Oh, I'm walking around. I'm lurking. I'm stalking. I'm walking around the room. I'm looking over your shoulder, and as I'm looking over your shoulder, what do I typically do?
Comment on things and thoughts.
Yeah, I comment on things that I'm seeing on your paper, comments on things that you're writing down, asking you questions to get you to think more deeply about those things that you're writing on your paper. Oh, have you thought about X or Y as you're going through and processing that? When I cut you guys loose to do something like that, and I'm stuck in my office and you're stuck wherever you are for that time period, just know that no one wants to be more in class than I do because I don't get to do that thing. I don't get to do that thing where I get to walk around and talk to you about the things that you're thinking in the time period when you're doing the thinking. And I really miss that about teaching with people in an actual classroom. So I'm going to try and find other ways of doing those activities to get you doing more of your own thinking and to keep me out of strict lecture mode, which in this environment I tend to fall into that a lot. But know that when I do that, it's like sheer torture on my side of the screen because you guys are busy doing something. And I would normally be busy doing something during that time also, but I'm stuck here not really being able to do anything. I haven't used breakout rooms in this class because it's a class of 5, and normally a breakout room would be about 4 or 5 people in a larger class. But if it would help you, like Elizabeth said, you know, it was different doing it on your own than being able to discuss the answers with you. When I do those exercises in the future, I'll break you guys out into breakout rooms, even though it seems odd to have a breakout room of 2 and a breakout room of 3. If you guys would find that to be useful when we do those things, so that's on my list of things to keep in the back of my mind. Was there anything else that I missed that were kind of big takeaways that you guys had for Judy? So what happens is she gives me a list of things, and since she doesn't know you, there's no names or anything attached to them. And basically, these are just kind of the responses that people in the class had. And then she and I talk about them for about an hour and a half, and then I then come and talk to you about it. Anything other major things that I missed that you can recall? Okay, all right. Today, what I want to do is I want to just finish talking about water relations. So from the standpoint of what you need to know and what I'm kind of expecting of you from this series of time periods where we're going to be talking about physiological ecology is amphibians and reptiles, like every organism, has particular physiological challenges that they have to overcome in order to survive out in nature. One of these is maintaining water balance. And so you have to balance water gain versus water loss. And organisms can do this morphologically by things like modifications of the skin. They can do this behaviorally by changing things like postures. And they can do things physiological, like doing things like generating metabolic water as one source of water. So the details that are coming out on each of the slides, they're kind of focused on that big picture idea of how do organisms deal with water limitation, or in some cases, overabundance of water, or in other cases, just the wrong kind of water. And we're going to talk about the wrong kind of water today somewhat. So hopefully you've read this chapter dealing with water relations. So we finished talking about metabolic water, and we were talking about waxy tree frogs, right? At the end last time. So somebody, while I pull up the PowerPoints, somebody remind me what the deal is with Phylomedusa, the waxy tree frogs.
Don't they? I don't remember exactly what it is, but they wipe something on them to help prevent water loss.
Yeah, so rather than mucus glands, they have lipid glands, modifications of the mucus glands, an evolutionary modification of the mucus glands. And they produce this waxy substance that takes the place of mucus, and because it's lipid-based and waxy, it actually forms a better barrier against water loss. And then as their skin glands secrete this, they wipe it around all over their body to get a uniform spread of this. And it's that waxy coating that then allows them to have a higher skin resistance. So we're talking about Chiromantis and Phylomedusa, these arboreal frogs that have wax glands rather than mucus glands, and then they spread this all over their bodies. So think about this from an ecological perspective. Why would this modification, these mucus glands evolving into lipid-based glands, why would this be the kind of modification that you would expect to see in arboreal frogs rather than a terrestrial frog? Because all these frogs that are in this group of waxy frogs, they're all tree frogs.
Terrestrial frogs might be more likely to do like estivation, whereas arboreal frogs are less likely to do that.
Okay, so one reason could be that terrestrial frogs can burrow into the soil. Soil is almost always more moist than the surrounding air. And they might also just have better access to standing water as well, might be another reason. But they can also estivate. They can go in and do what we're about to talk about, which is cocooning behavior. But when you think about a tree frog, what is a tree frog surrounded by constantly? Because it's in a tree. You're on a tree branch. What are you surrounded by?
This might be dumb, but leaves.
So you're surrounded by leaves, but leaves aren't really necessarily interacting with you in a way that's important from the standpoint of moisture. Those leaves are attached to stems. So here you see Phyllomedusa selvagei sitting on a stem. The only parts of its body that are in contact with the stem are really its feet. And otherwise, that animal is surrounded by what?
Like just air.
Just air. And the whole time that it's surrounded by air, what's happening to the liquid in its body?
Evaporating.
What's that?
It's evaporating.
It's evaporating. Yeah, it's just constantly evaporating. So they're in trees, which means they're far away from any standing water, because standing water just generally doesn't occur in trees for the most part. And they're completely surrounded by air. Many of these species live in the tropics, where you might expect it to be warmer. And so they're just constantly losing water. And so it makes sense that they would have some sort of adaptation, either modifications to the number of layers of the skin or the thickness of the skin, or in this case, the situation that has been evolved independently a number of different times is the modification of these mucus glands into wax glands, then put this waxy coating on the outside of the body so that it protects them from evaporation across the skin into air. And it's probably better for you to have evolved something like that than to have to be constantly climbing up and down the tree to go to a pool of water at the base of the tree. You can just stay in your environment where you're at and exist there, because you have this evolutionary modification that prevents evaporation, or another way of saying that is it increases skin resistance. And so you see that the frogs that have the highest skin resistance happen to also be these frogs that live in the tropics and are tree frogs in the tropics. So another way of approaching this kind of material as you're going through and learning is, so here's an adaptation to a particular challenge. What is it about the environment that this organism finds itself in that might have driven that particular adaptation to have occurred in this group of species in this particular environment? Elizabeth mentioned cocooning. Cocooning is something that occurs in a variety of different species. The example in your book is the African, and I have spelled that incorrectly, the African bullfrog. There's no such word as African. The African bullfrog, can somebody explain to me whether it's the African bullfrog or whether it's the spadefoot toads that live in the desert Southwest, what's going on with cocooning in these groups? The picture on the left is an African bullfrog that has been excavated from its burrow that is still encased in its cocoon.
So I think it's when they secrete this mucus, and like when the mucus hardens, it hardens with the multiple layers of skin, like shed skin, and then it creates like the hard barrier between the frog and like the outside soil.
Yeah, so what it does is it burrows down. Once it's underground, it does exactly what you said. It secretes this mucus, but this mucus is trapped underneath the outer layers of the skin that it normally sheds periodically. If you've ever kept an amphibian in captivity, you will oftentimes see in the aquarium that it's in, you'll see these flocks of stuff floating around in its water, and that's just the shed skin. So they shed their skin in ways that are analogous to the way reptiles shed their skin. But in this case, the mucus gets underneath those old skin layers, and as that mucus fills up, it causes the body of the thing to kind of balloon out, and it's basically layers of old dead skin with mucus underneath. Those outer layers harden up as the environment around the animal dries out, and then that serves as this kind of water impermeable barrier, such that the thick mucus and the hard outer coating prevents water loss, further water loss from the frog, and they can spend, in some cases, months underground in these cocoons. This is something that typically happens in amphibians that live in seasonally dry environments. So there's usually a protracted dry season followed by a somewhat limited wet season. In the desert Southwest, the wet season comes on with the monsoon rains that occur typically in the fall of the year. And you can see that pattern in some tortoise data that we'll look at later on today, hopefully. What then happens as the rains come and water starts percolating down through the soil? So you may have a toad or a frog that is a meter down in the soil. That's terrible drawing of a frog. It's in its cocoon. And when the rains return, rain is falling on the surface, but then slowly this rainwater will percolate through the soil, moisten the soil. As it moistens the soil, when it reaches the level of the frog, what starts to happen to that cocoon?
It dissolves.
What's that?
It dissolves.
It doesn't dissolve so much as it begins to soften. And so as moisture begins to build up in the soil around it, the moisture moistens those outer layers. Those outer layers begin to soften. As they soften, they become more permeable to water. As they become more permeable to water, what happens to the mucus inside that cocoon? If you put any mucus in water, what would happen to it?
Like it becomes less thick.
If you added water to mucus, would it become thicker or thinner? in terms of its consistency?
Thicker.
Who was that?
That was me.
Would you say thicker or thinner?
Thicker.
Yeah, it will actually become thinner because the protein that makes up the mucus actually just becomes more and more dilute as you get more and more water in there. So as the cocoon softens, water starts to diffuse into the mucus layers of the cocoon. That mucus becomes thinner and thinner, and the frog actually has the ability to detect the chemical changes in the mucus. And as that mucus becomes more and more dilute, the frog detects the chemical changes that are occurring in that more and more dilute mucus. And those chemical changes prompt that frog to basically exit estivation. Once it gets the neural signals that have picked up the changes in the osmolality of the mucus, that then kind of wakes the frog up. And once the frog is woken up, it basically starts moving around inside the cocoon. That ruptures the cocoon. And then once that happens, the frog is free to climb, to dig itself out of its burrow and come out onto the surface. Because the only reason that that set of chemical changes has started happening is because on the surface, there are these torrential rainfall events that indicate to this animal that it should come out of estivation, get to the surface, go to a nearby pond and breed, because all of the other frogs in the area are also exiting estivation and going to the pond to breed. And then they'll go to the pond and breed. After they have bred, they will go back to where they had dug their burrow previously in that general vicinity. They'll dig a new burrow and go down and start the process of estivation again. So there are chemical changes that occur inside the cocoon as that cocoon is being weakened by the encroaching water that is the signal that the frog is receiving from these torrential downpours that are occurring up at the surface level. And this happens with African bullfrogs. It happens with the spadefoot toads here in the Midwest and into the desert Southwest. This same process occurs. Questions about cocooning? Okay, so we've talked about water loss across the skin of amphibians and how this is more of a challenge for amphibians than it is for reptiles. But you also have to consider water loss across the respiratory surfaces. So in most frogs and salamanders, not only do you have the potential for water loss across the skin, but you also have the potential for water loss across the lungs. And that's true for amphibians, but then it's also true for reptiles. And so as you breathe in and breathe out, as you breathe air in, that air is generally going to be drier than the air inside your lungs, so that when you then breathe that air out, the air that you breathe out is moisture, and that moisture that is in that exhaled air is water that you've basically lost from your body via your lungs. And so organisms also have to be concerned about respiratory water loss. Tangential aside, I've been biking outside lately, and it's been windy. And that wind has been a warm, dry wind coming from the south. And one of the things that I've noticed as I have been biking is normally I like to take a drink when I first start biking, and then I try and take a drink every 15 minutes as I'm biking, because it's just a good habit to get into so that I keep my water balance up. And so I've gotten in this habit. Well, what I've been finding lately is because I've been in the presence of these dry winds coming up from the south the last couple of days, is I'm having to drink more often. I'm drinking every 10 minutes or so, simply because I'm feeling drier. But that feeling of dryness isn't because in the first 10 minutes I'm working out such that I'm sweating all of my moisture out. What's happening is I'm breathing more heavily because I'm biking, but because I'm breathing in this warm, dry environment, I'm losing more moisture through respiratory water loss from my lungs that I'm losing through the pores of my skin because I haven't even really started sweating. But about 10 minutes in, I need a drink, and it's all due to water loss across the lung. So in this way, as I'm biking, I'm in many ways like an amphibian or reptile, breathing in a dry environment. Hopefully the relevance of the tangential bicycle story is clear to you. So another form of water loss. I think this is the final form of water loss, is water loss through getting rid of nitrogenous waste. So your kidneys filter fluid from your blood, and not only does it filter out water, but it also filters out solutes. And so the urine will be full of nitrogenous waste that come as a result of the metabolism of protein. So we talked about protein metabolism way back when we talked about metabolic water. And so protein metabolism leads to nitrogenous waste buildup, and you have to get that stuff out of your bloodstream. And so your kidney filters water through the glomerulus. It's a little ball of capillaries, and there are thousands of them or tens of thousands of them in your kidney. And that's actually where the pressure of the blood squeezes the liquid portions of the blood out through gaps between the cells of the capillaries and the glomerulus. You don't need to know this. You don't need to know the ultrastructure of the glomerulus. Have you guys taken anatomy or physiology? Did you guys talk about kidney function in there? So those of you who've taken a physiology class will know about the structure of the glomerulus. It's basically just a ball of capillaries, and the blood pressure in the glomerulus is such that it causes little gaps between the cells of the capillaries and allows fluid to leak out. That fluid that leaks out is not only just water, but it's also water filled with waste products. And then the structure of the kidney is such that it concentrates those waste products. So it reduces the amount of urine that you have and makes that urine more and more concentrated in terms of the amount of the waste products that are in there. And depending on how your kidney works, it will basically filter those things out, and they will appear in the form of ammonia in a monotelic species. It will come out as urea in ureotelic species, or it will come out as uric acid in uricotelic species. So there's this correlation between how concentrated the urine is and how toxic it is. But then that is also correlated with how much water is required to get rid of that stuff. And in the case of monotelic organisms, these are organisms that excrete the nitrogenous wastes primarily in ammonia. It's energetically cheap, but it's toxic. And because it's toxic, it requires higher levels of dilution by urine. And so a monotelic, it's energetically cheap to metabolize proteins in a way that leads to nitrogenous waste that are ammonium, but that ammonium is toxic, and as a result requires more water to dilute it. And so you excrete a lot of urine. How many of you have ever picked up a toad? Found a toad in your yard and picked it up? Okay, Kiara and I are the only people who've ever picked up a toad. That's sad in a way. Kiara, what's the first thing the toad did to you when you picked it up?
He peed on it.
It peed on you. And not only did it pee on you, it probably filled your palm with pee, didn't it?
Yeah.
Because it has a very large bladder, because it's creating a large amount of urine, because it handles nitrogenous waste by converting them to ammonia. That ammonia is toxic, and as a result of that, it dilutes that in a large volume of urine to reduce the concentration of it. Ureotelic produces urea. This is what we do. It requires more energy to produce nitrogenous waste as urea, but as a result, it is less toxic. Because it is less toxic, it requires less dilution, so this is a more water efficient way of getting rid of nitrogenous waste. When you secrete your nitrogenous waste as uric acid, it requires a lot more energy than producing urea, but the advantage of this is that it's not water soluble. And so you don't actually need any water to excrete it. And it's also not toxic. It's basically inert. It occurs as these crystals that don't really do anything. And so the amount of energy required to produce ammonia versus urea versus uric acid goes up. As that goes up, toxicity goes down. But as toxicity goes down, the amount of water required to dilute that and get rid of it goes down. So the type of nitrogenous waste that you produce is directly correlated to how much water you need to lose in the process of excreting those nitrogenous wastes. Questions about kidneys and nitrogenous waste excretion? Okay, the last thing that we need to think about are situations in which organisms find themselves in an environment that causes them to experience blood osmolality levels that are too high. Do you guys remember from chemistry what osmolality is? What is osmolality? You didn't think you would need to know any chemistry in herpetology class, did you? When you think of a solution, what are the two parts of a solution? This should be easy, going back to the very basics. What are two parts of a solution?
Water and the salts.
Okay, so in this case, we might think of it as water and salt, since we're talking about things like salt glands, but basically this is the solvent and this is the solute. The solvent is the liquid portion, and then the solute is this stuff that is dissolved in the liquid portion. And so osmolality is a measure of how much solute there is dissolved in solvent. And so as the concentration of solute goes up, osmolality is going up. So if you have high osmolality, it means that you have a lot of solute dissolved in a small amount of solvent. And so organisms that have this difficulty generally occur in two places. They're organisms that live in salt water, or they are organisms that live in places where you're subjected to protracted periods without a source of water. And so you can experience increases in blood osmolality due to the fact that you're not bringing in any water while your metabolism is building up solute concentration in your blood flow. In either of these cases, that exists as a problem to the organisms that are experiencing that. So we'll handle organisms that live in salt water first, and then we'll handle organisms that live in deserts next. So anytime you have a source of salt that you can't avoid, you build up salt in your system. That builds up salt in your blood, essentially, the salt concentration in your blood, and you have to get rid of that. One way of getting rid of that is actually through the kidneys, but oftentimes your kidneys aren't really designed to get rid of salt as much as they're designed to get rid of nitrogenous waste. And so organisms that live in salt water will have what we call salt glands. And the organisms that we're thinking of are mainly the sea snakes and the sea turtles. So the sea snakes are in the family Elapidae, and the sea turtles are in two families, the Chelonoidae and the Dermatochelidae. And the location of these salt glands on the body are really diverse, and oftentimes they are either modified salivary glands or tear glands. So if we look at salt glands, we find salt glands do occur in terrestrial organisms. So Dipsosaurus is the desert iguana that occurs out in the deserts of the desert Southwest. If you were reading Coles and Bogert, you saw them referring to Dipsosaurus as one of the species they were working on. They also worked on Uma. Uma scoparia is a species of fringe-toed lizard in the desert Southwest. But Hydrophis is a species, is a genus of sea snake. Caretta is a genus of marine turtle, sea turtle in the family Cheloniidae, and then Crocodilus. This is actually looking at the saltwater crocodile in Australia. What do you notice about differences between terrestrial organisms with salt glands and marine organisms with salt glands? And this is a image looking at the concentration of ions in the salt gland secretions.
Does it have something to do with the potassium concentration?
It does. So if you look at the terrestrial organisms, Dipsosaurus and Uma, what do you notice about their potassium levels in salt gland secretions relative to the marine species? Go ahead, Kiara, you started down this path.
Well, the concentrations of potassium are higher than in this. Are the ones on the right hand side the sea ones?
Yeah, Hydrophis, Caretta, and Crocodilus are all marine organisms. Dipsosaurus and Uma are terrestrial.
Yeah, so there's a more concentration of potassium than there is in the sea animals.
Yep, and in the ones that are marine, the salt glands are mainly getting rid of what? Somebody else. Quinn.
They're getting rid of salt like Na and Cl.
Yeah, they're getting rid of, what you normally think of as your typical salt, sodium chloride, which is the salt that is found in ocean water. And so it makes sense that the type of salt that different organisms are getting rid of is correlated with the type of environment they're finding themselves in. And so in this case, you're dealing with the influx of salt into the organism as a result of being surrounded by salt water, seawater constantly. Here, you're getting a different mixture of ions of salt because you're dealing with increases in osmolality due to a lack of water availability in the environment. These are both desert species. And so we'll talk about adaptations to deserts here in a minute. So I mentioned before that the salt glands are derived typically either from tear glands or from salivary glands. And so in the case of Dipsosaurus dorsalis, you see that the salt gland is actually a modification of a tear gland located up here in the head around the eye. The nasal gland, the lacrimal gland of Caretta caretta is once again a modified tear gland that has been modified into a salt gland. But in the case of Hydrophis and Crocodilus, these are modifications of salivary glands that are modified to expel salt. And in the case of Hydrophis, it's a big solitary gland. In the case of Crocodilus, there are an aggregation of lots of smaller glands located on the tongue that are responsible for expelling salt. And so a diversity of locations and a diversity of independently evolved types of glands that have evolved from what was their original purpose, which in one case was was secreting tears, in the other case secreting saliva, and they've been modified for secreting salt. We talked about earlier in this talk about water balance, we talked about water postures that were designed to gain water, but you can also have organisms exhibiting water postures to prevent the loss of water. And so when we go back to these water postures, like the snakes and the ant-eating lizards of Australia and North America and tortoises, these are all water postures that are designed to increase the amount of liquid water that an individual gains. But we can also have water postures that are involved in conserving water by preventing water loss. So this is an example from your book. This is Eleutherodactylus coqui, a frog species, a small frog species that lives in Puerto Rico in the rainforest there. In Puerto Rico, there is a wet season and a dry season. And so it is possible that these animals do experience times when they are prone to losing too much water from their bodies across these respiratory surfaces, which are the skin. And A and B here are water conserving postures. And you can see that the animal has tucked in all of its legs. What it's basically doing is it's reducing the surface area that is exposed to air so that it's creating the smallest surface area to volume ratio. You'll recall in the Coles and Bogart paper, they talked about the surface area to volume ratio in terms of heat gain and heat loss, but the same thing can happen with water loss. And so one of the physiological ecology things that is important is the surface area to volume ratio. And a perfect circle has the lowest surface area to volume ratio. And if we take a circle of a certain size and a certain volume and basically express that in terms of surface area that is arranged in a rectangle, we find that we fit inside this rectangle much less volume relative to this surface area. So the surface area to volume ratio goes up as you deviate from this spherical pattern. And so what you can see when an animal is trying to conserve water is it tries to roll itself into a ball. These two individuals here are in a severe water conservation posture and a less severe water conservation posture, but all of the rest of these frogs are in different postures. These are more alert postures. This is actually a calling posture. And in a calling posture, the animal has raised its body up off of the leaf. It's standing up on its forward limbs and its hind limbs such that there's air going all the way around the body and there's air going around all of the limbs as well. So this animal, because it's in this calling posture, is exposing the most possible surface area to the air. And so it has a lot of surface area relative to its volume. This has the least amount of surface area to the volume in figure A. C, D, and E are definitely not trying to conserve water. A and B are definitely trying to conserve water by reducing the surface area that is available for the evaporation of water across the skin. And this is a type of tree frog. It's in the family Eleutherodactyli, but it lives in trees. And it is an arboreal species that does not have modifications like wax. And so it's subject to water loss across the skin more so than a wax producing frog would be. And this is the end of water. So we talk about osmolality, and this is basically osmolality increases as we talk about the concentration of solute, in this case in blood or in urine. And this is the case of the desert tortoise. And so what happens in the deserts of the Southwest is that there's a monsoon season that comes late in the summer and into the early fall. And this monsoon season is due to global patterns of air circulation. But after those monsoons come, you go through a protracted period of time when rainfall in the deserts of the Southwest are relatively low. And so if you're a desert tortoise, you have access to a lot of free aquatic water, liquid water during these time periods. And the rest of the year, you're getting water primarily through water found in your food. And so what happens to these animals is that they become more and more dehydrated as time goes on throughout the year. But what happens to the blood of the animal is at the beginning of this long protracted time period, the plasma osmolality is at a particular level, and it stays at that level pretty well throughout the entire stretch of the dry season until the very end of the dry season when it begins to creep up. And that's ultimately bad for the animal, because what that means is that it's getting more and more concentration of solute in its bloodstream relative to the amount of solvent that is there. But what's happening in the urine that is in the bladder is the urine starts out at a relatively low concentration, and then as you move into the summertime, move into the dry season, that osmolality in the urine creeps up, and then it also creeps up here at the end. So what is happening with the situation in tortoises during this protracted dry period, when they don't have access to liquid water, they basically store urine in their bladder. They basically start out the season with a bladder full of urine, and then they proceed to not pee the entire dry season. And what they do with the urine that is in their bladder is they basically move the water components of urine out of the bladder and into the bloodstream. What that does is it allows the bloodstream to maintain osmolality at safe levels while concentrating osmolality in the bladder. So as you move water out of the bladder into the bloodstream, osmolality in the bladder increases until you finally have concentrated osmolality in the bladder as much as you can. And at that point, you start to then get increases in osmolality in the bloodstream because you can't draw any more water off of the urine that is in the bladder. When the rains come, blood osmolality falls and urine osmolality falls because finally the organism has access to liquid water. It starts drinking. As it starts drinking, its kidneys add more water to the urine. And at that point, the animals begin to evacuate that urine and build up more urine for the next dry season. That urine then serves as a source of water to keep blood osmolality levels within safe ranges until the next dry season comes along. So this is one particular way in which an organism in a very dry environment can cope with drought by basically storing urine. The nice thing about your bladder is that you can concentrate that urine in the bladder, and it doesn't have negative effects for the rest of the body because the bladder is this self-contained unit. But you can continue, in tortoises at least, to draw water off of the bladder in order to translocate that water to your blood supply to keep your blood at healthy levels. And that's a pretty cool little adaptation to drought, protracted droughts in the desert Southwest. Questions about water balance? Friday, we will start on temperature. We will begin talking about temperature by looking at the Coles and Bogart paper, and that will kind of set the stage for how we think about thermoregulation in amphibians and reptiles, but in reptiles in particular. So come into class on Friday prepared to talk about that paper as a group, and then we'll move on to the material that's in the textbook once we've kind of hashed through that. Questions? All right, then I will see you on Friday for lecture, and I will see you tomorrow in lab, and you'll continue working through the reptiles.